national webinar is Advancement of Nanotechnology in the Treatment of Reproductive Tract Diseases, a Journey from Bench to Bedside. The advancement of nanotechnology in the treatment of reproductive tract diseases marks a significant leap from laboratory research to clinical applications. Nanoparticles have revolutionized drug delivery by enabling targeted therapy for conditions such as endometriosis, reproductive cancer, and sexually transmitted infections, improving efficacy while minimizing the side effects. Additionally, nanotechnology aids in early diagnosis through biosensor and advanced imaging technique and is being explored in fertility treatment and hormonal regulation. While challenges remains in safety, regulatory approvals, and accessibility, these innovations promise transformative solution for reproductive health, moving closer to personalized and effective patient care. So, looking at the challenges and research potential in the field of reproductive tract diseases, this webinar took its inception. Today, the virtual seminar hall is full of positive scientific vibes because of the presence of the eminent guests and participants from various parts of the country who spared their precious time and graced the occasion with their benign presence. Thank you so much. So to begin with today's national webinar, I would like to invite Professor Pradyumna Mishra sir, member ISSRF Executive Committee, to please welcome and introduce Professor N.K. Lohiya sir to deliver welcome address and brief us about ISSRF. I also request Manisha madam to please share sir's profile slide. Thank you, sir.
Good morning, everyone. Hello, good morning. Are you able to hear me?
Good morning, sir. Yes, sir.
I consider this as a rare honor and privilege to be amongst you this morning, I am extremely thankful to Professor Neelesh and Professor Dinesh Kumar Mishra for having accepted to both of you for having bestowed me the responsibility of introducing President ISSRF, Professor N.K. Lohiya sir, a doyen in the field of molecular reproduction and development, and he is considered a father figure in the field of reproductive. So if I start explaining to you about the accomplishment achievements of sir, I guess it would be beyond the school, because since the slide already projects sir's academic accomplishments, so it is of words and please, sir, the floor is yours, sir. Thank you so much. Namaskar.
Big thank you, Professor Pradyumna Mishra. And since your long association with me, particularly, and with ISSRF, has been a matter of great pleasure. And I appreciate your initiative of today's event. And I also compliment particularly Rahul Nahata Saab, because when looking into the profile of the scope, that is Smriti College of Pharmaceutical Education, I saw that this institution was established in 1999, not very old. It's only 25 years of its establishment. And then I saw that Professor Neelesh Malviya, who being the principal of this, and joining hands is Professor Dinesh Mishra, who has been earlier associated with the ISSRF in a number of ways. and he made a lot of contribution to the activities of the ISRF. And earlier associated with the, I could see other distinguished people joining this. So before I proceed, I must the activities of the ISRF. Background voice other distinguished people joining this. So recording. Okay. I must compliment the organizers, particularly when I saw that a large number of delegates have registered. It's more than 1,000. It means I saw 1,121. And interestingly, it has also been spelled out that who all have joined, including professors, doctors, Mr., Mrs., and students. So I convey my greetings to all those who are linked virtually with this through the Google link, or they are at YouTube links and other media links, and they are participating in this event. And I'm sure that listening to very distinguished speakers, which have been identified by the organizers, they will be greatly benefited. I take this opportunity to briefly convey what is ISSRF, and the reason being that many of the students who have joined might not be aware about the society, about its activities, about its mandate, and so on and so forth. Starting with this first slide, you see that the society was established in 1988, and this was Dr. T.C. Anand Kumar's initiative through which the society was formed when he was there as director, Institute for Research and Reproduction, Mumbai. And subsequent to that, it was nurtured by subsequent presidents of the society. And currently we see that it has taken a very big platform. And now we see that it not only disseminates information in the form of the newsletter, it also in the form of the journal, and it has almost more than around 1,700 life members of the society, and you could see and appreciate that it has a large number of medical colleges, universities, nursing homes, clinics, research centers, diagnostic labs, and pharmaceutical companies and including funding agencies who are part of this. When we see next, as I said, that the society has its own official publication in the name of Journal of Reproductive Health Medicine. And journal is published by the scientific scholars from US, and we see that it is very regularly publishing, and I have great pleasure to invite all those who are attending this webinar to please contribute to the success of this journal. Recently, we lost our very distinguished scientist in the name of Dr. V.P. Kamboj. I hope and I wish that everybody who is attending this is aware about it, that he was the director of CDRI, Central Drug Research Institution, and those who are pharmacy students, pharmacy faculty, they must be knowing Dr. Kamboj's immense contribution. And he was very closely associated with the ISRF for last number of years, or I would say from the very beginning, 1988. And for every event, his contributions have been immense. So society thought, why not we bring a special issue of the journal in great memory of this very distinguished scientist. And again, I'm very pleased to share with all of you that it is Professor P.K. Misra who initiated this. And he said that, sir, he most willingly agreed to shoulder the responsibility to bring the special issue. And I am very happy to share that it is coming up well. And the scientists who are associated with Dr. Kamboj and those who are associated with the reproductive health research, they are contributing. And we wish that before the 2025 event, we would be able to come out with this special issue of this journal. Then, in addition to this journal, which of course makes a very good visibility of our society, but we also have the biannual publication in the form of ISSR newsletters. And the society has published so far 32 issues of newsletters. And you would be very pleased to know that covering all different aspects of the priority health issues are being taken. And all these newsletters are available on the website of the ISSRF. And those who are interested, they can have the PDF form copy of any newsletter to go through. And this newsletter, once published, we provide it on the website of the society. We send it across to all members of the society, about 1,800. And then also we send it to all premier institutions of the country in the form of soft copy and also hard copy. So also available in their respective libraries. Again, you could see in this slide that there are two newsletters in pipeline. One being edited by Professor Samir Kumar Patra. He is one who is a professor in the Institute of Technology, Roorkee, and being supported by Professor P.K. Misra and Professor Rupesh K. Srivastava from AIMS. And he's editing newsletter on epigenetics, mechanism, and reproductive biology, and it is coming up well. And we look forward its compilation by the end of this year, and make it available to all during the upcoming ISSRF 2025. The second newsletter, again in pipeline, is being edited by Dr. Gyanendra Singh. He is again in the ICMR institution, in Institute of Occupational Health at Ahmedabad. He has been also closely associated with the activities of the ISRF, and he is talking about occupational hazards affecting reproductive preventive measures to mitigate the risk. And I am sure that these two newsletters, with the great initiative of Professor P.K. Misra, will be available by the end of this year. Then just a glimpse. I just want to share with pleasure that our society is very vibrant. I am making this statement based on its activities. We have regularly, without any fail, annual meeting of the society ever since it was founded. We have midterm activity of the society, and even today's event is also part of it. And recently we had another activity on 28th, 29th at Pilani with the Balika, Birla Balika Vidyapit, very well organized event. And these, some of the photographs which indicate that either in annual meeting, which you see that even the ex, at that time he was the Director General, Indian Council of Medical Research. So the what you call governors, the eminent scientists from different institutions, the DG ICMR, and all that have been part of our ISSRF. And you would be pleased to learn that even the young students from different institutions who are exposed to awareness programs of reproductive health, they also very immensely contribute to the success of our ISRF activities. And sometimes we see in Pilani, where you see that they make beautiful rangoli, and rangoli which is a expression of their talent, covering different aspects of reproductive health, and we saw that this is greatly appreciated by all who attend such meetings. Then today's meeting, well thought title, theme is commendable. It is first time I saw that the midterm activity is being addressing the reproductive tract diseases. We know that generally we have been talking about reproductive health in totality, but reproductive tract diseases forms a very important part of reproductive health. And talking about the nanotechnology, which the honorable principal of this esteemed scope conveyed, that how important is nanotechnology. We see that this nanotechnology advancements and dealing with the reproductive diseases, this is being likely and is to be addressed by very distinguished identified speakers. So I compliment the all who are associated with this, and this is being listed starting from patterns, as I mentioned earlier, Sri Narendra Nahata Sahab, Rahul Nahata Sahab, and convener Neelesh Malviya, and Dinesh Misra, and Pradyuman. Why I am mentioning is the driving force behind of the society. Then it may not be proper to talk about nanotechnology, what it is, for the type of audience, because it is the pharmaceutical group, and nanotechnology is very close to them. So I would just simply skip this slide, but you could appreciate that what it is, and measure your life in nano is being expressed that what is the relevance of the nanoparticles and how they can be reflected. If we see how nanotechnology is, it has application, we see it has application in every discipline, be it a bioengineering, be it a cosmetics, and be it a optical engineering, nano devices, medicine, drugs, formulations, nanotechnology, nano fabrics, everything, we find that it has a implication. If we look into the nanotechnology and reproduction, this slide is being prepared by Dr. Barkha. I must acknowledge Dr. Barkha also for her inputs in various ISRF activities. And we see that how beautifully it is being demonstrating that how reproductive health, as mentioned, starting from gamete formation to the fertilization, to implantation, to gestation, to parturition, to anything and everything. So we say prostatitis, endometriosis, ectopic pregnancy, erectile dysfunction, infertility, and so on and so forth. It is being listed. And all these things related to reproductive health being addressed by nanotechnology. And you see nano detection, the dendramides, the ligand-based targeted nanoparticles, and all these different formulations, they are helpful in addressing various reproductive health issues. And this is the nano you see it in the future, which we see that nanotechnology has a great future in not only addressing reproductive health issues, but in a broader perspective about drug formulation, treatment modalities, everything. I have the privilege to extend invitation to all of you. You would be pleased to know that our society is going to have its annual meeting, and this is in the name of ISSRF 2025. And this meeting will take place in Jaipur during February 14th and 16th, 2025. And this is being organized by Dr. Bilal Institute of Biotechnology and Dr. Sudipti Arora is the organizing secretary. And the venue is the best venue of the state of Rajasthan in the name of Rajasthan International Center, where this event will take place. And you all are cordially invited to join this. Then this is, as for ISSRF is concerned, last but not the least slide to show you that if you click ISSRF, simply www.issrf.org or ISSRF, you will reach to ISSRF various activities in the past, current, and future. So I encourage all of you, when you have time, please do it. And again, I must say it is Dr. Barkha and Dr. Ansari who are contributing, ensuring that timely the society website is updated. And what is happening day before yesterday or yesterday, that is reflected in the website of the society. That is a commendable thing which is happening. Next slide. Yeah, this was last slide. Okay, so I'm sure that as for my part of conveying about the ISSRF is completed, but I would say that this was a glimpses of the society, and I would be more pleased to listening to very distinguished speakers being listed in today's event. And I would be very pleased to see that the association of the scope is not only limited to today's activity, it is likely to continue, and we will see that we will have a strong bonds in times to come. And I would like that scope and all associated with this event becomes a part of ISSRF in times to come. Big thank you for this opportunity, and compliments to one and all for contributing to today's event. Thank you.
Thank you, sir, for your kind words of wisdom and blessing us and introducing us with ISSRF. It is really a proud moment for all of us that the society is giving its scientific contribution since 1988. And we all would feel proud to be associated with this society in the times to come and willing to publish our research findings in ISSRF journal and newsletter. Sir has also enlightened us on the topics of nanotechnology and its application in reproductive health. Thank you so much, sir. Moving ahead, I consider it a great honor to welcome our tireless and dynamic principal, Dr. Neelesh Malviya, sir, for the welcome address and opening remark about the webinar.
Thank you, sir. Am I audible?
Yes, sir.
Yes. Excellencies, distinguished delegates, ladies and gentlemen, it gives me enormous pleasure to extend to you all a very warm welcome on behalf of Smriti College of Pharmaceutical Education, Indore, to convene this national webinar 2024. And the theme of this national webinar is Advancement of Nanotechnology in the Treatment of Reproductive Tract Diseases, a Journey from Bench to Bedside, under the auspices of the Indian Society for the Study of Reproduction and Fertility. Scope: Smriti College of Pharmaceutical Education, Indore is a premier institute of Central India, imparting pharmaceutical education in a quality manner to stir out technically competent pharmacy professionals. If we talk about our today's national webinar theme, conceptually, nanotechnology has brought about a significant transformation in global healthcare and is expected to impact clinical medicine shortly. The use of nanomaterials shows great promise in addressing various gaps in reproductive healthcare, including the diagnosis of cancer, fertility preservation, and the treatment of conditions related to puberty, menopause, sexually transmitted infection, and human immunodeficiency viruses. While Numerous clinical trials have been conducted using nanoparticles to detect cellular disorders and target drug delivery. Most of these initiatives are still in their early stages. To effectively translate this advancement from the laboratory to practical clinical applications, the need for collaboration between disciplines such as pharmaceutics, pharmacology, pharmacochemistry, natural chemistry, pharmacognosy is paramount. Each disciplines play a crucial role. Collaboration is essential for integrating nanotechnology into clinical practices. A strategic partnership combined with a comprehensive role in bridging the gap between translation research and product development by addressing regulatory and technical obstacles, we can facilitate the successful integration of nanotechnology based tool and technology into clinical practices. We are indeed elated and obliged to have such venerable and illustrious personality as our eminent guest and resource person in today's webinar. At this point, I would like to extend a warm welcome to our distinguished guests, delegates, principals, various resource persons, technical experts, all the participants. Firstly, to grace the occasion, we have with us Professor N.K. Lohiya, sir, President ISSRF, a well-known personality. Professor Lohiya is Emeritus Professor NAMS, NASI, Senior Scientist and Secretary NASI Rajasthan Chapter. We have with us Professor P.K. Mishra, Scientist F and Head Department of Molecular Biology, National Institute for Research in Environmental Health, Indian Council of Medical Research, Kamala Nehru Hospital Building, Bhopal. So please accept my heartfelt thanks for mentoring us to organize today's national webinar. We have with us Professor Dinesh K. Mishra, Professor, Department of Pharmacy, Guru Ghasidas, a Central University, Bilaspur. Sir is renowned personality in the field of academics, having more than 2 decades of expertise in academics and research experience. He is known for excellence in research. Sir contribution to plan this national webinar deserve a big round of applause. Thank you very much, sir, for your unconditional support to organize this national webinar. If we talk about our resource person and technical expert who accepted our invitation to deliver their expert talk, we have with us Dr. Shashank K. Singh, Senior Principal Scientist, Cancer Pharmacology Division, CSIR, Indian Institute of Integrative Medicine, Jammu, to add applause to the inaugural. Our unfailing gratitude to Dr. Tathagat Datta, President and Chief Technical Officer, Zodas Expoin Private Limited, Hyderabad, and Dr. Vaibhav Dubey, General Manager, Kashiv Bioscience LLC, Vadodara, to have concentrated the national webinar with his earned appearance. I would also like to extend a warm welcome to Dr. Neelesh Malviya, Assistant Professor, National Institute of Pharmaceutical Education and Research, Naipur, Hyderabad. Your acceptance of our invitation is most graciously acknowledged, sir. I would like to welcome all the exalted speakers in the national webinar. My heartiest welcomes to all the principal, especially my true friends and mentor, Professor Dr. Sujit Pillai, sir, Dr. Sapna Malviya, all the faculty members of SCOPE, and all the senior authority of Mansoor organization, Mansoor University. Our heartfelt proclaim to our patron, Shri Narendra Nahata and Shri Rahul Nahata. Their reflective abilities have highly motivated us, and without their supports, it's not possible to organize this type of national seminar. Thank you very much, sir. And now may I extend my thanks and appreciation to our members of the organizing committee and program committee for continuously working on this national webinar organization. Today we have around 1,400 students from various colleges from entire country and also from pharma industry. Now, on behalf of the organizing committee, I am pleased to declare this. I would also like to extend a warm welcome to open this ceremony, this inaugural ceremony of national webinar. Our wish is that you will enjoy this. I would also like to extend a warm welcome to contribute effectively towards it and take back with you knowledge, experiences, good connections, networks, and the contacts definitely will help in your future research related to nanotechnology in the connection with reproductive diseases. Thank you very much. Have a happy memory of this national webinar. Thank you very much. Thank you.
Thank you, sir, for your rich words. and brief us about the latest scientific development in today's theme and introducing us with today's eminent speakers. Moving ahead, we are very fortunate to have amongst us Dr. Shashank K. Singh, sir. So I request Dr. Dubey madam to please introduce our next speaker and also request Mrs. Manisha Dire madam to display sir's profile slide. Thank you.
Thank you, sir. Dr. Shashank K. Singh, sir, is presently working as Senior Principal Scientist in Pharmacology Division at CSIR, Indian Institute of Integrative Medicine, Jammu, India, in the area of cancer drug discovery from the last 20 years. IIM Jammu is one of the esteemed laboratory of CSIR, Government of India, which engaged in novel drug discovery program from the natural, semi-synthetic and synthetic sources. Earlier, he has worked with Win Medicare Limited R&D and its sister R&D organization, Modi Mundi Pharma Research Development Center, Switzerland, in Formulation Development Department and in Process Validation in Cadila Pharmaceutical Limited, Dhaka, Ahmedabad. He has done his postdoc in year 2009 to 10 at Medical Center, Ohio State University, Columbus, USA, and PhD from Delhi University, India. He has earned his master's degree in pharmacy from Birla Institute of Technology and Science, Pilani, Rajasthan, India. He did his bachelor's degree in pharmacy from Dr. H.S. Gore Central University, Sagar. Sir has more than 100 international publications in peer-reviewed journals and 6 US and European patents to his credit. He has been awarded as Indo-US Fellow in 2009 to 10 by DST Indo-US Science and Technology Forum, New Delhi. His areas of interest are cancer drug discovery from natural products, drug combination studies, targeted drug delivery systems for cancer, and cancer immunotherapeutics. We would like to welcome him for an insightful thought and to deliver keynote address. Please, sir.
Yeah, thank you very much. Am I audible? Hello?
Yes, sir, you are audible, yes.
Yeah. Thank you, Dr. Kushagra. Thank you very much for nice introduction. At the outset, let me share my slide first. Is my slide is visible to all?
Yes, sir.
Yes, sir.
So thank you very much for giving me chance to present myself in this August gathering. I am first of all, at the outset, I would like to thank the organizers for giving me this opportunity. And I'm thankful to Professor Dinesh Mishra, Professor Pradyuman Mishra, Professor Lohiya, and all other organizers present over in this meeting. Today, I will be talking about the nanotechnology in female reproductive disease and diagnosis. As already Dr. Lohiya was explaining that how nanotechnology is changing there is a paradigm shift after discovery of these new technology, and with the advent of new technologies in the nano field, we have seen tremendous or unprecedented changes in the overall outcome of the disease, whether it is related to treatment or diagnosis. So first of all, let us see what are the diseases which female are basically affected with related to reproductive organs. Like common diseases are endometriosis, polycystic ovary syndrome, ovarian and cervical cancers, uterine fibroids, and infertility. So these are one of the most common diseases which are usually occurs in female, and early diagnosis is overall improves the overall outcome of the treatment. So how the nanotechnology is helpful in these reproductive as a reproductive medicine and detection of these diseases. So if you see the nanomedicines, if you see in terms of delivery of these drugs, nanotechnology play a very critical role. As Professor Lohiya was explaining, that 0.1 nanometer to 100 nanometer scale, which is basically a nano size. So if we play in between, there is altogether change in the overall characters physicochemical property of a compound or drug. So we can we can increase the surface area of the drug, and we can increase the exposure time. So by changing the size of a material, we can make a very advanced material which can be utilized for different purposes. So these delivery system, whether it is a micellar kind of structure, whether it is a dendritic structure, or whether it is a multilayer liposomal structure, or there are different forms of nano formulations. So we can deliver the drugs through transmembrane and through liposomal accumulation, and by releasing these drugs to the target site, we can increase the overall outcome of the drug. So I will be explaining in the later slides that how these nanomedicine delivery systems can impact the overall outcome of the treatment plan. Nanomaterial is also, you have seen that nowadays so many biosensors are available to detect the disease at the early stage. So these biosensors are made up of nanomaterials, and they play a very critical role in they have a supervisory role to control the disease, whether it is located in the central site like brain, uterus or a blood stream. So these things can be monitored using these bio nanosensors. So they play a very critical role, like luteinizing hormone or follicular stimulating hormone. Level of these hormones can be measured by utilizing these biosensors. So because you know that these hormones, they play very critical role in female diseases like estrogen, progesterone, follicular stimulating hormone, luteinizing hormones. So by maintaining their levels, or by measuring their levels through biosensors, we can have a very we can have a selective treatment plan, or very personalized treatment plan for individual. So nano reproductive technologies either can be used for assertive assistive reproductive technologies like IVF, and so they can be used for contraception also. These technologies are very helpful. So what is the major drawback in these diseases? Because most of the diseases, whether it is ovarian cancer or cervical cancer, the stage at which we diagnose these diseases is almost like stage 3 or stage 4, and then it becomes very difficult to treat these type of cancers, because they are already in advanced stage of disease, and they have like from local disease to they have reached to the advanced stage, like they have proliferated to the other organ or tissues. So it becomes unmanageable to treat these type of diseases. So how this nanotechnology can play a very critical role by monitoring and diagnosing the disease at the early stage. So what are the difficulties we usually face? What are the challenges in diagnosing the female reproductive diseases? Like there are some invasive techniques like biopsy, laparoscopy, and but these are all invasive techniques, because if somebody is diagnosed with cancer, the doctor will ask him to go for bone marrow biopsy. They will take out the bone marrow tissue from the bones, and they will evaluate if there's some blast cells or other cells are present in the so they will whether he is suffering from leukemia or other diseases. So bone marrow biopsy is one of the biopsy. Then incisional biopsies are there, needle biopsies are there for breast cancer, and sentinel node biopsy is for lymph nodes. We check that whether there is any cancerous cells are present or not. There are some non-invasive techniques also being used clinically, like ultrasound, MRI, and hormonal test. They also give information about the status of disease, whether this disease has been there, or there is occurrence of disease, or it has spread to the other organs. So by monitoring the disease, we can use these technologies. But how nanotechnology is changing the diagnosis part, I will just explain you. So yesterday I was just going through that, because I am not a domain expert in the reproductive biology. But since Professor Dinesh Mishra asked me to talk about this disease, so I just gathered some information that how nanotechnology is useful in this area of research. So basically, what happens is, in the advantage of nanopore technology, you have heard that in the past last 3-4 years, we have seen the genome sequencing has taken place in the most of the diseases, like now the disease, there is a paradigm shift in the drug treatment. Now most of the cancer are being treated through a personalized treatment, like one has to give his blood sample or tissue sample. They will see.
Dr. Shashank,
yeah
the slides are not changing actually. You are still at the very first slide.
Okay, shall I share once again?
Yeah, it's not moving.
1 minute. Can I see the slide? Now the slides are changing.
Yeah. So can you put it in this slideshow mode?
Yeah. So now is it moving?
Not really. No. स्लाइड शो में करके एक बार
बंद हुआ था।
So now can you change?
Still I am on the first slide, or you can?
Yeah, you on the first slide. Yes.
Now is there any change in the slides?
No,
No, sir.
जो कर्सर को नीचे लाइए आप एक बार एक एक करके देखते हैं स्लाइड शो में बिना Yeah, it was moving. Yeah. अब आइए नीचे. Yes. Yes.
But it is not in power full mode. Yeah.
That's fine. That's fine. That's fine. If
Okay,
that's fine.
so
We are able to see
I am
now.
really sorry for that, because they are not moving. So I was on this slide actually. I was just explaining you the nanopore technology. Is it visible advantage of nanopore technology?
Yes, sir.
Yeah. So basically, this technology is based on nanotechnology, because there are, if you see our biological system also, there is nanopores, and the material or drug that get transported through these nanopores, which are basically present on the cell surface. So these nanopores or ion channels like structures, they are in size of 0.1 nanometers to in that range. So they basically, Professor Beamer in Harvard University, he took, it is a very old technology. 1935 they started this technology, and around 2015 they came with this idea that these nanopores can be utilized for delivering for sequencing the DNA. Because you have seen in the code during COVID times, we have seen that whenever there was new variant like Omicron, or there are new variant of corona came, first thing they used to sequence the genome of the coronavirus, and then they will get the sequence, and then on doing the checking on the reference genome available on the PubMed website, they will come up with that what type of variant it is. So based on that data, they will declare this is Omicron virus, or what type of coronavirus it is. So similarly for personalized treatment also, this technology is very helpful. Like this is based on nano platform, where they have used the alpha hemolysin kind pores. So there they have a silicon plate, and above that there is a making biological membrane, double layer bilipid layer is there, and from that nanopores there is beneath the silicon plate, there is a fluid flowing throughout. Like it is just like a water is flowing, water current is flowing through a pipe. So if it is passing, so DNA, once it enters into this ionic flow, so basically a mild current is passing through that flow. So if current is, we know that DNA is negatively charged molecule, so if we have a positive energy there, so it will pass through that pore. And if there is change in the ionic current, that will be measured with the help of different detectors or biosensor which are fitted in the chip. So it is a very small palm size equipment, and which can be used. Oxford Nanopore is the company which is basically manufacturing this equipment. It is a palm size, so anybody in field, or if he is in field or anywhere, he can do the genome sequencing just connecting with the laptop computer. He can just get the sequence of genome of a virus, or any RNA containing virus or DNA containing virus, or for that matter any biological material. He can do entire. And the beauty of this machine is like it can, you can have a long read sequences also can be measured, and the depth is also very good. So by doing this genome sequencing, you will come to know that what type of mutations are present in the disease. Or likewise, if we know that the BRCA1 mutation, breast cancer gene, is basically responsible for all breast cancer, and there is high chance of getting ovarian cancer also if BRCA1 mutation or BRCA2 mutations are present in the genome. So you can easily sequence the genome by taking just a small isolating the DNA and making the library, and by using these library you can just have a sequence of the genome. So it is very fast as compared to traditional methods. It is very accurate for detection of genetic abnormalities. It is very affordable. Like entire read can be done in a one go. It is a real time this thing. So what I was explaining, just you can see over here that it is a real time, high resolution molecular detection, label free, high throughput screening. It analyzes DNA, RNA, protein, small molecule. So you can see that there is a motor unzipped double stranded DNA is there. So the unzipping is done by the motor protein which is present on the surface. So these proteins, pores embedded on the membrane surface, and DNA, single stranded DNA is passing through these pores, and the base pairs which are attached to the single stranded DNA will give a different ionic current when it passes. So these detectors will have signals picked up by the blockade of the current caused by the nucleotide passing through the pore, which can be give like ATGC, different type of nucleotide will be there. So they, once they will pass through this nanopore, they will deflect the current and they will block the current, ionic current. So you will have different signals, and those signals can be measured through a detector. So by this technology, you can see this is miniature device of the machine, which is just like a palm size, pen drive size of machine, where you can just load your DNA material, and just it can be used bench to bedside, like any places. If you are roaming, if you are traveling, or if you have to collect samples from field, you just need a high speed internet connect, high speed computing GPU kind of machine. Along with this, you can just sequence the genome, and you will have a real time monitoring of disease progression. So with the advent of these nanopore, like nanotechnology has played a very definitive role in this. It can be used in point of care diagnosis of disease at different locations. And it is as I've already told, is thumb sized device and USB. It contains 512 channels, and per channel there are 4 ports are available. So on these ports are utilized for sequencing purpose, and these are the consumable items. After once they are saturated, these has to be replaced. So likewise, the sequencing can be done very easily by these nanotechnologies. You can see that there is deflection in the current. So ATGC has been measured. So these motor proteins, they just unwind the DNA, unzip the DNA, and only single stranded DNA pass through these nanopores, and that can be measured. So utilizing these nanoscale devices, the translocate polymer molecules in a sequential monomer order through a very small volume of space, include detectors that directly convert characteristic feature of translocating polymer into electrical signal, and transduction and recognition in the real time on a molecular by molecular basis. It can probe thousands of different molecules in a few minutes. It can probe very long lengthy DNA sequences also, because there are different type of machine like RNS. There are different sequencers are there in the market, but for getting the long read sequences, they have very limited number. Like for doing sequencing, you have to first fragment the DNA in small parts, and then you can make the library, and then you have to attach with the adapter proteins, and then you can go for sequencing. Whereas the beauty of this machine is the entire long read can also be done, because entire sequence can be passed through these nanopores. So application of this can be applied for epigenetic changes. Like we know that in most of the hypermethylated state of the DNA can lead to various mutations or change in the DNA repair mechanism. So these type of epigenetic changes can be monitored by using these nanotechnologies, like BRCA1, BRCA2 mutations, breast cancer causing, and so these mutations can be easily identified. Hello, am I audible?
Yes, sir.
My slides are changing.
Yes, sir. So can you please?
So identifying these epigenetic changes in endometriosis and polycystic ovary syndromes can be done through these machines. So I will just take a case study of ovarian cancer. You know that nanopore sequencing detect the early stage ovarian cancer, identify BRCA mutations for risk profiling and early treatment. So if somebody has diagnosed with, if there is BRCA1 mutation, so there is high chance that in the near future or early stage of life, she can develop a breast cancer or ovarian cancer. So these type of cases for early detection of the disease, these technologies are very helpful. So we can also utilize the, you know that CA125, this cancer antigen 125 is a well-known marker for detecting the ovarian cancers. So these biomarkers can easily analyzed by these machines. And there are some new technologies like nanoparticle based photoacoustic imaging, which are ligand functionalized proteins or nanoparticle can be used for measuring the acoustic imaging. NP based fluorescence imaging can also be done by utilizing the CD44 which is a proliferative marker, and estrogen receptor mediated fluorescent imaging can also be done. Then you have other supramagnetic nanoparticles, which are used as a contrasting agent for diagnosing the cervical cancer. So these are also useful nanotechnologies which can be utilized for detecting the ovarian and other type of female cancers. So how the nanotechnology in treatment of female reproductive is useful. So there can be different type of nano carrier which can be deliver the desired drug to the organ or desired site. There can be nanomicellar structures. There can be carbon nanotubes. There can be lipid bilayer nanoparticles. There can be nucleic acid like siRNA kind of uh, nanomaterials. There can be amphiphilic random copolymers. There can be zinc oxide or silver or gold nanoparticles, pegylated nanoparticles, liposomes, antisense delivery systems. So there are different type of nano carriers. I am not going to talk about all those delivery systems because you all are expert in that area. All are from pharmacy background. You have studied in your text that how these delivery vehicles are useful in delivering, or how we can functionalize these delivery vehicle for deliver target specific drug delivery or for exosome deliveries. So if you talk about the ovarian cancer treatment, it is one of the most aggressive gynecological cancer, and usually it get diagnosed at the late stage of disease. And because of absence of early symptoms, the conventional treatment usually is the mainstay of the treatment lies on the surgery and chemotherapy. And but there is limitation due to toxicity to the healthy tissues, and because of drug resistance, uh, sometimes it becomes very challenging to treat advanced stage ovarian cancer. And nanotechnology plays a very critical and promising role to improve all treatment plan and reducing the overall side effect in the late stage or advanced stage of disease like stage 3 or stage 4, where the survival rate of the patient is very limited. So conventional chemotherapy usually given for patient who is suffering from ovarian cancer is paclitaxel and cisplatin, which have systemic side effects like neuropathy and damage to the healthy tissues, and they develop sometimes drug resistance also. So the patient who are refractory to these type of treatment of paclitaxel or cisplatin, so how to treat those patients has become very challenging task. So the nanotechnology has given the solution for this problem. Because of targeted drug delivery, we can ensure the chemotherapeutic agent can be delivered at the target site. We can control the release profile of the drug. We can sustain the release profile by managing the pharmacokinetic behavior of the drug. By reducing, we can also increase the patient compliance by reducing the frequency of dosing, by improving the overall compliance. And one of the classical example of this is like liposomal doxorubicin, which is commercially known as Doxil. And Doxil is one of the success story of nanotechnology based therapy for ovarian cancer. It is a liposomal preparation of doxorubicin, and it is basically encapsulated within the liposome, spherical vesicle made up of lipids, which allow the drugs to circulate in the bloodstream for longer and extended the half life of the drug. So you know that doxorubicin is a very toxic, and one of the major toxicity is the cardiotoxicity, which because of which uh, these nano formulation help in controlling these toxicities. So targeting tumor cells, liposome are designed to accumulate in tumor tissue via enhanced permeability and retention effect, ERP, which we call ERP in our pharmaceutical field. So this EPR effect is very well known. Because of this, they get retained because of small size, because the blood vessels of tumor are highly permeable. So these nanomaterial get accumulated in these blood vessels, large gaps of these tissues, and which allow the doxil to preferentially accumulate in these tumor site and have a sustained release at the target site. And by doing this, we can reduce the overall side effect of the drug of traditional doxorubicin with a significantly we can reduce the cardiotoxicity of the drug. So, which is also ensuring the safety profile of the drug. If you see the formulation of doxil, which encapsulated in the bilipid layer of phospholipid and cholesterol, and have a pegylated polyethylene glycol. Size is around 85 to 100 nanometers. So if you see the preclinical data of doxorubicin, in comparison to the adriamycin or a standard drug without a formulation, and if doxil is given, so 6 mg per kg of body weight doxil has been given to the animals, and if you see the adriamycin also given in the same level, and the saline, you can see the mean tumor volume has reduced drastically. If you see the comparative data with adriamycin as a without formulation, so it clearly indicate that there is clear cut benefit of this bilipid liposomal preparation of doxil in reducing the overall tumor volume. And rationale for exploring ovarian cancer is like it is superior to adriamycin. It is basically indicated for the patient who are heavily treated or pre-treated with cisplatin like drugs, or paclitaxel, because they have become refractory. Their tumor has become refractory to the patient. They are not responding to the treatment. So those patients are being advised, or patient who have already been at the advanced stage of disease are being given such kind of formulation. So there you can measure the response of CA 125 has also gone down, and tumor shrinkage is more than 50% of in the case, and overall quality of life is also get improved by this treatment. So clinical studies of patient with platinum resistant ovarian cancer who received doxil show improved outcome compared to those treated with conventional doxorubicin. Improved progression-free survival. Patient treated with doxil experience longer period with without cancer progression. Reduced toxicity. We can reduce the overall toxic side effects like alopecia or gastrointestinal disturbances. So that can be reduced significantly, and patient can have a better quality of life with these kind of formulations. So if you talk in terms of cervical cancer, which is also one of the reproductive disease in females, and worldwide cervical cancer is one of the fourth most common female cancer worldwide, and comes under top 3 cancer affect the woman under the age of 45. So worldwide cancer, cervical cancer has high frequency. And if you talk in terms of Asia also, India stand at 5th position in the world, like Indonesia, Myanmar, Brunei, Mongolia, and India. So there is high prevalence of this disease in Indian female population also, and high mortality rate is there. So what is the, we all know that the human papilloma virus is a causative virus for this disease, and majority of cases of cervical cancer results from the infection with the human papilloma virus. And the human papilloma virus DNA identified approximately in the 95% of malignancy or malignant lesions in the cervix of female. So there can be two subtypes of this HPV 16, HPV 18, which majorly cause cervical cancer and vaginal valvular cancer, and non cancer type of tumor also HPV 6 and HPV 11 cause, which 90% of no genital warts type of tumors, but they are non-malignant tumors. So the confirmatory test followed by PEP's test, you know that PEP colposcope is used to check the sign of cancer, and punch biopsy is also used for confirmatory diagnosis of cervical cancer. Cone biopsy is also used. Doctor take a deeper layer of the cervix for testing. So HPV, if you see a HPV, a small virus which has 8,000 base pair, non-enveloped, double standards, DNA circular, this thing. And HPV double standard is circular DNA genome, and genome has a fragment integration. So HPV genome expresses transcriptional program encoding E1, E2, E4, E5, E6, and E7, which regulate the viral life cycles. So cervix cancer is caused almost exclusively by high-risk human papillomavirus. The human HPV genome encodes two oncogenes, E6 and E7, via mechanism that promotes host genome instability, and virus integrate into the host genome cells. So basically it get E6 or E7 get integrated into the host genome, and then they start giving the oncogenic signals. So long read nanopore sequencing, again the what I have explained you in the beginning, that same like here also long read sequences can be read through nano sequencer, and where cervical cancer study of cervix, because these HPV integration can be complex. The host HLA gene are often somatically mutated in cervical cancer, and this region and any variant are difficult to resolve in short reads technologies. So here also the advantage with this technology, because it can do the long reads also. So short read becomes difficult to identify the piece which has been integrated into the host genome. So the somatic mutation which has taken place in the cervix can be difficult to analyze with the other sequencer, whereas the nanopore technology has got better advantage, which can do the long read sequences also. So HIV transcription and splicing are incredibly complex mechanism. So HPV infection begins in the basal epithelial of cervix. If you see in the picture, once the virus enters into cervix cells, it replicates into nucleus and as a circular double-stranded DNA called episome. So this episome start multiplying, replicating, and virally virus assembly. These episomes again start infecting to the other cells and their DNA material. So these virus oncoprotein E6 and E7 are expressed from the episomes. These inhibit the host tumor suppressor genes like TP53 and RB1, which are basically cell cycle regulators. So they suppress these tumor suppressor genes and disrupt the cell cycle in the cell line, in the cells. And HPV may also integrate into the host genome, leading to amplification and overexpression of cellular oncogenes and genome instability, facilitating oncogenes. So by integrating the piece of DNA E6 and E7, they just take over the cells and they start giving oncogenic signals. To resolve this complex sequencing process, large scale structural variant and long and ultra long nanopore reads can be done, and full phase integration event into haplotypes directly methylated, because hypermethylated state will also, the epigenetic changes which are happening in the cells can be monitored by using this technology. Combining chromatin conformation captures 3C methodology is long in the long reads of nanopores can be utilized for identifying this disease. This reveal that there is integration of viral genome into the host genome. So the last thing is like endometriosis is also one of the common cause. The endometriosis is a chronic condition, you know, that where tissue is similar inside the lining of endometrium uterus. It is just like a lining of endometrium, but growing outside the uterus. So it causes severe pain, inflammation, fertility problem to the females. And traditional treatment for endometriosis include like GnRH therapies and surgery, but the outcome is not very good and very limited, and reoccurrence chance of disease is very high. And most of the females which take these hormonal therapy also face a lot of side effects like mood change, hot flushes, weight gain, bone density loss. So so these nanomaterial can also play a critical role. Like we know that our day-to-day life, or in our Indian cuisine also, we take turmeric in a large quantity. So turmeric is also a well-known anti-inflammatory compound. So these type of nanomaterials of curcumin can be utilized for treating the endometriosis by controlling these type of inflammation in the female body. By they are devoid of they don't have these side effects like hot flushes, mood swing, and bone density loss, as in the case of hormonal therapies. So they also control these therapy suppresses estrogen production also, androgen lesions also. They reduce the endometriosis lesions also. And surgical treatment, we know that there is very limited success with these type of surgical treatment, and they are give a temporary benefit to the patient. So, and there is high risk of scar formation tissue in the surgery. So these nano material of curcuminoids or other anti-inflammatory agents can be very useful for treating these endometriotic lesions. And one area of focusing of nanoparticles as a drug delivery, minimizing the systemic side effect of these drugs, can be useful option. So researchers are developing polymeric nanoparticles for delivering these kind of anti-inflammatory drugs directly to endometriotic lesions. And these nanoparticles are designed to release the drug slowly over a period of time and provide sustained relief from the pain and inflammation caused by the endometriosis. And functionalized nanoparticle can also be a useful strategy. Receptor mediated functionalized nanoparticles can also be utilized for delivering these type of drugs. And we can control the release profile also of the medicament by administering these type of nanomaterial to the patient. So there are some preclinical data is available in animal models for these type of curcuminoids, which reduces the severity of endometriotic lesions, minimize the overall side effects, and they have got good anti-angiogenic property, because you know that uterus bed is full of blood vessels, and it gives lot of fuel to the formation of new blood vessels, also give lot of fuel to this fire. So by cutting off the supply of these angiogenesis, or by cutting off the supply to these tumor like structures, we can just stop these ectopic growth in the female body by just administering these type of nanomaterial to the patient, and minimizing the inflammation in the body. So curcumin therapeutic effect significantly enhance reduced lesion size, and it can be effectively administered as compared to oral administration, because you know that curcuminoids usually are not bioavailable as such. If you give to the patient, because of poor bioavailability, there are different type of biocurcumins are available in the market where we can have better bioavailability of the drug. So these nano formulation can play a very critical role by increasing the overall bioavailability of these kind of anti-inflammatory agents, and which are used in our traditional system of medicine, and which can control the overall disease and minimize. So other approaches of endometriosis can be like iron oxide nanoparticle used for imaging endometriotic lesions in the animal studies. Liposome-based drug delivery systems for hormonal treatment, like GnRH and antagonist, are also being used. For infertility and contraception purposes, also like assistive reproductive technologies, we have seen that there is advanced materials are available where we can, which can help in for in vitro fertilization. Also it can be in IVF also, they can enhance the success rate. And nanoparticles for hormone delivery also, they have been there are many research papers are available where we can deliver the hormones at the target specific sites. So precisely we can say these nanotechnologies are going to be very useful for for in the case of infertility and contraception. So what are the challenges of treating these female reproductive diseases? That like diagnosis is one of the limitation. We know that most of the disease which at the late stage we come to know that there is advanced stage of ovarian or cervix cancer is there. So screening, lack of screening tools are available. So because of in advanced country there is a routine screening of patient, but whereas in India, lack of awareness and other things, we don't screen on regular basis. So because of that, we come to know at the very later stage of disease, and it becomes very difficult to manage the disease at the advanced stage. Systemic side effects of conventional treatment, we know that most of the drugs which are in the shelf are devoid of lot of side effects, and how to control these nanotechnologies can be very helpful for controlling the side effect of the drugs by increasing the overall bioavailability of the drug and making the drug available at the biological site where we can minimize the systemic side effect of the drug by utilizing the EPR effects and other, we can treat such kind of conditions. There are limitation with these technologies because the signal to noise ratio of most of these equipment are not very good. They are very uh, even though they are very accurate in diagnosing, but still there is scope of improvement because of low signal noise ratio and throughput level of the machines. Integration of artificial intelligence is very important for because without machine learning and other databases which are already available, because nowadays most of the hospitals they are collecting the data, and by utilizing this data with artificial intelligence, we can equip our population with a better diagnostic profile and better accuracy of diagnosing the disease at early stage. And there is still future of improvement in the reproductive health diagnosis. There are ethical considerations are also, like safety issues are there with the nanoparticles, like accumulation of the nanoparticle inside the biological system, and that can lead to a lot of toxicity. And there are regulatory challenges are also there. Stringent norms are required to regulate, ensure the safety of these nanomaterials. And future direction would be like it has got a great promise, but there are long-term effect of these nanomaterial, and still we need to understand in a great detail. And still investigation is going on in the most of the advanced biomaterials which are being used in human health. and nanomedicine often more expensive to develop as compared to traditional therapies. So still, cost cut is a very important aspect for cutting the cost of the product. We need to develop the good methodologies which can bring down the cost of the product. However, ongoing research and clinical trial continue to explore the potential of nanotechnology through more personalized treatment, like biosensors and all these things which can make the treatment more personalized. By using the genome sequencing, we can make the treatment more personalized to the patient. So we need to have more advanced nanomaterial, biocompatible material like silica and graphene. We need to have a more personalized treatment utilizing the nanotechnology, pave a way for individualized treatment. And there is still clinical trial going on and exploring the nanotechnology role in the reproductive treatment options. So at the end, I would like to say the nanotechnology has a lot to offer, a solution for early diagnosis of disease, effective treatment of female reproductive diseases, but yet continued research will yield a more advanced and less invasive treatment. And nanotechnology-based treatment like Doxil represent a significant advancement to fight against ovarian cancer. With continued research, we can expect more effective and safer therapies in future, and ultimately improving the overall outcome of for patient with female reproductive diseases. With this, I close my talk. I will be happy to answer your questions. Thank you.
Thank you, sir, for enlightening us on female reproductive disease and diagnosis and the role of nanotechnology in the treatment of diseases. Thank you, sir.
Thank you very much.
Thank you, sir. Now we will take a break of 10 minutes, and after that, we will begin our first technical session of Dr. Tathagat Datta, sir. Thank you all. We will be back by 10 minutes.